Imran's Raj. You know a lot of food because you work on brain and everybody else put it down. Anybody else has thought of brains? What really happens in brain? Have you guys thought about it? What happens? Okay, go ahead. Yeah.
Most fascinating thing about our brain is, till date, despite extensive research on brain, and till date, right now, scientists are working day and night to just study the brain, normal functions. All the normal functions of brain are not yet known. Again, one important thing is brain undergo rapid changes, and that phenomenon is called as neuroplasticity. Means whatever we people will expose, our brain will undergo the changes. So that is the main reason this brain disorders are incurable till date. These are the two most fascinating things.
Great. Neuroplasticity is an amazing thing, right? Very plastic. We are adaptable. We are adaptable individuals. We're adaptable animals. But that's very important, right? That makes us who we are. I can put you in jail for 2 months. You'll adapt to that environment. I can put you in a planet, in a planet. I can put you in an island, right? And you'll adapt yourself. Somebody talked about Martian, somebody talked about other movies, right? We adapt, which is a bracing, neuroplasticity. We adapt to environment. You might be in a toxic environment, you'll be adapted to that. You might be in a great environment, you'll adapt to that, right? Good, thank you. Neuroplasticity is one of the most fascinating things. We don't understand. As Mr. Raj says, we don't understand these things at all. It's very difficult to study. Brain is very difficult to study organ. Why? Why did I make that statement?
Because it is like our brain is very complicated made up of different things, and there are certain neurons which are working for that. Some neurons are working in that way, like someone's brain is working from left side and someone's from the right side. So that is very fascinating that left brain and the right brain working differently in.
Okay. Yeah, great. What do you see on screen? You guys know about these? Parietal lobe, temporal lobe, brainstem, frontal lobe, cerebellum, occipital lobe. I don't remember these names, by the way. I just put it up there. Okay. They have different functions, which is brilliant. Different parts of brains have different functions. We have neurons, we have oligodendrocytes, we have glial cells. What else do we have in brain? What are the major cell types? I say 3: neurons, oligodendrocytes, glial cells. What else? Huh? Astrocytes. Okay. Microglias. I thought they were similar, right? Okay. Good. They're important for different functions. The functions of our daily life are as such what makes us who, or if our physiological functions are differently distributed in different parts of the brain. It looks very similar. I'm not going to get into the brain, of course. I just wanted to sort of emphasize that different parts of the brain likely have different processes, just same as my eye has a different function than my intestine, or my gut tube. My intestines have a different function than my liver. Is different than my stomach. Brains, different parts have different functions. Therefore, what must be important? Coordination. Okay. Why am I talking about this in an epigenetics class? And I told you guys on day one, why do we study epigenetics? Yeah, that's right. So what I want to point out is different parts of the brain have different functions, therefore different genes must be expressed in a different way. So the epigenetics of different parts of the brain might be likely different. I don't think people have figured that out still. Okay? But it is possible that there are different parts of the different epigenetic functions in different parts of the brain. Okay? and maybe I don't know enough, maybe some of it is figured out already, so don't quote me on it, but I have not seen literature where they say, okay, parietal lobe has different, epigenetic marking or substantially different epigenome than temporal lobe. Maybe we've not looked. Maybe there are different kinds of epigenetic elements. Maybe there is, just like I said, there's CPG versus non-CPG, right? Maybe people didn't look at that in the right way, right? Maybe we have not looked at all those other 150 histone modifications that we talk about, right? Because they're not there normally. So people have figured out that the epigenetic profiles in the brain are fairly different from the rest of the body. Okay? That's why this field is very fascinating. And again, very few people work on neuroepigenetics. It's very, very limited area. Most of the review articles I try to read even now, like this week, to try and understand what should I teach, are very, 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 very preliminary. Meaning, they just say DNA methylation changes on the genes. That's what I'm going to be telling you in the next few slides. It's all about DNA methylation changes on this genes, this genes, this genes. That's it. Nothing more than that. Not deep enough biology there. There's an opportunity for us to better understand how does different epigenetic functions might lead to different roles that these different parts of our brain play. Okay? Please do think about that. That's very important. So there's a lot of emphasis now in the world in research of brain, okay? Because we want to understand, right, who we are. Because we figured a lot of other parts out. I mean, I make that statement loosely, right? But brain's really very untouched in a lot of ways because very difficult to study. Neurons are neurons, you believe, and a lot of this function is very, very dynamic. And most of the things we do in other organs are static, right? I take a liver today, if I'm just normal, I take a liver tomorrow, I'm not going to see much of a difference, okay, or a different cell type. But brain functions are within milliseconds, right? And that is where the problem is. That is why the research is always lagging because, I mean, in terms of regular, functions of brain, right? Of course, you can think of neurodegenerative disorders, etc. That's likely going to be fairly constant over some time, and you could do that. And people have done that. That field has come up in last 5 years or 10 years at best. Okay, but really on a daily basis, like how you respond to emotions, how you respond to a specific cue, etc. Very difficult to know what's going on. Maybe epigenetics doesn't have a role to play, or may not be enough, because you don't want all this, 50 things coming in, changing the transcription of one versus other. Again, I'm making you think, okay? I don't know enough about this field either, but I'm making you think as to why one would think that we don't know enough about these. Why have we not seen differences, right? What have people not looked? So you must, when you decide on to work on something, must think, what's the problem? What is my bottleneck? Bottleneck is what I solve, right? Because as a scientist, you solve problems. What are problems? Problems are things that are bottlenecks. Same, right? So if you have to first define the problem in doing any research, so and then you have to have right tools to be able to do research, right model organisms, right technology, right, to look into things. But having said that, I mean, there's substantial research in the field, so we'll talk about that. Okay. All right. I did tell you earlier that non-CPG methylation happens, right, in brain. Interestingly enough, when you look at the evolution of how this methylation occurred over time, and I'm going to talk about methylation because that's what mostly is known in the neural field. There's not much known otherwise. Okay. As it evolved as this non-CPG methylation evolved in different organisms, there was evolution of MBD4 and MECP2 expression levels. Do you know what is MBD4 and MECP2? We talked about this, right? They're methyl binding proteins. MECP2 is a very important methyl binding protein. I will talk about that in a second again. MBD4 is also a very important protein. I mean, it was very close to my heart when I did that work that I showed you earlier. MBD4 has an MBD domain and a DNA glycosylase domain. These two proteins are different than the other MBD proteins by just structure, okay? And they're different from each other too. But this is what came about in terms of evolution. As evolution happened, these things evolved, okay? And that's when you say that, okay, these things must have a function because evolutionary logic applies, right? We got something at some point, a trait or a protein or a gene that was needed at that point of evolution, right? And evolution happened over billions of years, right? So evolution is a great tool to think of new biology as to what might have happened at that point. CRISPR, somebody's interested in CRISPR. CRISPR's evolved as that host defense systems evolved in bacteria, right? That's how they were discovered. CRISPR evolved to just kill DNA for the foreign object, right? So these are great tools. You always must think of evolution. That's why I said earlier to you guys too, right? Don't think no God, no nothing, not one Adam Eve appeared on one day or nothing like that. We did evolve, right? At least that's what the science tells us. We believe in science, right? We all evolved from the organisms. We all evolved from lower organisms. So you must use that logic in your research as you go along and think about what was the evolutionary logic behind this, and that gives you a lot of insight. We ignore it. I mean, we don't think about it on a daily basis. We don't ignore it, right? So that's one. I just want to give you that lesson to think about. So then people have figured out roles of these proteins now, and I'll talk about that in a second. In brain function, in brain methylation. Okay, so that's important. Again, uh, I think this just talks about evolution when that happened and the brain CPH methylation happened. This particular motif evolved, which was non-CPG. And that's where then they know that MECP2 and this proteins work. Okay, all right. In the neurodevelopmental systems, of course, we all evolved. Our brains evolved as we go from obviously a zygote to a fetus, and fetus to adult human brain. This matures over time, and it matures, in a systematic way. But a lot of that maturation involves the panel B on the picture, which is essentially differentiation of neural stem cells into different types of cells. And we know in differentiation, epigenetics plays a fundamental role, right? Epigenetics plays an absolutely key role in most cell differentiation because remember, neural stem cell had one piece of DNA that got into a neuroblast or an OPC or an astrocyte or an oligodendrocyte or a glial cells. They made their different cell types all together, their different functions, right? And those different functions then obviously evolved because of epigenetic reprogramming that allows the change in transcription of a large number of genes. Okay, cool. Indeed, people made knockout mice for DNA methyltransferases. DNMT3 and 3B together. That's a double knockout mice. There are other knockout mice as well. There's a conditional mice with somebody called nestin-Cre. Nestin is expressed in the neurons. Nestin-Cre. Do you guys know about knockout mice at all? The genetically engineered mouse models, right? So you can engineer the embryonic stem cells with specific vectors that would have Cre lock system. Do you guys know about Cre lock systems? No? You guys don't know about Cre lock systems, right? Cre is a kind of a recombinase that works on these lock sites. And when Cre binds to the two lock sites, it's going to delete the region in between. Okay, is that clear? I don't have a slide for it, but you can read about it if you want. This is a very, 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 very useful tool that is used extensively in biomedical science. Cre locks system. Cre recombinase binds to lock sites. Whenever Cre is expressed, it will bind to the two sites, it will bring them together, it will be homologous recombination happen, and the site in between will be deleted. So what people have done to genetically engineer them is, let's say I want to delete DNMT3A or DNMT3B. I would express Cre in that system wherever I want to, okay? And that Cre, and I will put the two lock sites surrounding one of the exons of DNMT3 or 3B, which if was deleted, it will make a non-functional protein. It will make a premature stop codon so that the protein never gets made completely. Get it? Right? That's how most of the genetically engineered knockout model systems have been made. Cre-lock system allows you to make conditional mice. What does conditional mice mean? Meaning you want to delete a gene specifically in a given organ or a given cell type. How you do that is you take the tissue specific promoter. In this case, it was a nestin promoter. Nestin is only expressed in neurons, right? Get it or no? Oligodendrocytes. I think it's oligodendrocyte, if I'm not wrong. Okay. Now, but anyways, nestin is expressed. We put a Cre gene under nestin promoter control. So Cre will only be expressed where nestin is expressed. Okay. So let's say a gene, any promoter, you can take any tissue specific promoter. People have made hundreds and thousands of mice like this. So I can take a promoter that is specific to my retina, okay? And I can just put my Cre under that control of that promoter. So that gene will only be expressed in the retina, So my Cre will only be expressed in the retina. Okay, got it? And then I will delete wherever my lock sites are only in the retina, right? Same way they did it here in the oligodendrocytes, for example. Okay, they delete it only in the neuronal systems. I'm sorry, I'm kind of confused. Wherever, doesn't matter. Nestin Cre, nestin expresses Cre. Lock sites are again around DNMT3 and DNMT3B, so it's going to delete both those genes. Cool? We got this? Yes or no?
Yes.
Confused? No. Read about genetically engineered mouse models if you're interested. There are tons of videos out there. Okay, genetically engineered mouse models. You can use genetically engineered zebrafish models, genetically engineered anything. Okay, genetic engineering is just genetic engineering. I am a genetic engineer, right? If I just put a gene somewhere, I'm a genetic engineer. Okay, got it. So when you delete DNMT3 and 3B, it, well, not in a normal circumstance, I'm sorry, DNMT3 and 3B methylates these neuronal genes which are written here, and that gets suppressed or activated, and then the neuronal differentiation occurs. When the knockout happens, these genes are not expressed. This is a reverse function, and then there is an abnormality in the differentiation. So DNMT3A3B is important for neuronal maturation. Okay, and that's important because neuronal maturation will control your behavior, your memory, your learning, everything. Okay, so therefore this has an impact, and I'll come to that. An experiment done in this paper later. Why don't I come to that now before I go in there? Okay, in this particular model, when they knocked out DNMT3A3B specifically in the neurons, okay, they saw the defect that I just told you about. There is defect in maturation, and here they did a learning experiment. Okay, let me show you a small video on this. It says something called Morris water maze. Are you guys confused? You guys look all confused to me. Are you guys confused? No? Sure? Everybody is on page? All you did, all I explained was just to tell you that there is a knockout, DNMT3A3B knockout, specifically in the neurons, and that when you delete it, you change the neuronal differentiation, if you will. Okay, or oligodendrocyte differentiation. Cool. I want to show you a video, just for a minute or two. Where is that? Craziness. Okay. Oh man, I forgot. Where is the video? Morris water maze, right? Morris water maze. Okay, let's just show a 2-minute video for you guys. You can see my screen after a minute, of course.
We provide forward-thinking leaders and innovators with the products they need for groundbreaking research. Over the years, we've been at the forefront of innovation, empowering science.
Move down, move forward, move forward. Okay.
Where they must swim to an forest water maze. Animals are placed in pools of water where they must swim to an escape platform. During training, the platform can be exposed about 1 inch above the water. The animal must learn to swim to the platform and remain there to be rescued from the pool. After the animal is trained, the water is increased and made opaque with milk or non-toxic paint, so the platform is hidden. The animal must then rely on memory to find the escape platform.
Hi, my name is Joseph Nunez. I'm a faculty member in the neuroscience program at Michigan State University. Hi, my name is Joseph Nunez.
Okay, that's what I want to show you for a second. Okay, basically, you have a mouse that you put into the water, and you put a target where it's going to survive. It's going to be there. It will survive. The life stick kicks up, right? So they essentially deleted the gene 3A3B, then subjected the mice to Morris water maze experiment. Okay, which you just saw. They did it for 3 days. It's saying here for 3 days they did the acquisition, meaning training the mice, right? Where the thing is, and then they did the reversal to try to now this is based on his memory. Okay, so he's going to go back and figure out where the thing was, and I need to go back and check. His first 3 days they put it in the same spot. You see, and then they put it on a different spot, and they leave it on a different spot. So that is learning, not memory. Learning that, okay, I need to go back to that. Okay, that's a simple experiment. Now you can see when they knock out mice, they actually take a larger path, meaning they're not sure, so their learning is defective compared to the wild type. So you're looking at blue over red. Okay, and here same way, latency means how long they took to the goal. All right, so their path was longer, they took longer to reach to the goal. All right, and time in quadrants. Okay, they spent how much time in each quadrant, I guess, and that's what it shows here. So the knockout was much longer, meaning knockout was defective. It did not have as well good a learning, right? What does it mean? The DNMT3A and 3B are critical for, or very important at least for learning or learning behavior. What do DNMT3A and 3B do? DNA methylation. I just told you, people have shown that DNMT3A is critical for that non-CpG methylation that we're looking at. Okay, so when you knock out the gene, this is what happens. Cool? All right, and those, as I said previously, will modify the gene methylation on specific genes. So memory, there was learning. Okay, where are we? Here. And similarly, people have done experiments in memory. Okay, and this is more going to be a summary, and there's from another paper where they talk about the phase of memory. Memory is here, what they do is they put the mice in a cage, they give them a loud response and a poke in their feet. Okay, the poke in the feet will be a memory. Okay, I got, this is a problem. Then they'll consolidate, they'll retrieve the mice. Now they'll give only the sound and no poke. Okay, so then the mouse differentiates, okay, that he's got that the sound is not a problem, poke is the problem, because that's what gives me the pain, right? And then this is the experiment is done, though, and this is our memory experiments. Typically people do it. Again, I'm not an expert, you can read more about it. I just wanted to bring this to you guys to think about. Okay, that's an experiment people do. And then you say that these are different phases. The naive phase, encoding, meaning you're giving them the stimuli, then consolidation phase, then you get retrieval, meaning you're just telling them what was really the problem. So the memory of the problem is poke, not the sound, right? So they do that. I mean, this is probably very standard in the memory field. Again, people who do this all the time. And then the reconsolidate, meaning, just leave the mice alone, not do anything else. And then the extension of the fear. This is a fear memory, okay? Right? Because when he's pain, it's a fear, right? That's in amygdala. That's. So basically, in these experiments, people have figured out, and this is just a summary of several papers, and this came from a review article, that you have effects of link RNAs, which is a non-coding RNA that you will read about tomorrow, M6 RNA methylation, we talked about M6 methylation today, histone variants and our 3D structure, histone modifications in this process. And there are different people who have sort of, they showed the role of change in these patterns, just change in the pattern, not necessarily a causative pattern, okay, in these processes. Make sense? So I want you to know that there is epigenetics role in learning and memory. So our brains actually do require these epigenetic modifiers. All right, that's all I wanted to get you to think about, and you can read more if you're interested. We'll go up forward, back to what we had. Okay, I was here, and I just wanted to do that because I showed you the knockout mice earlier. The point of showing you that was in the normal neural differentiation processes of different kinds, DNA methylation, methyl transfers are important. Okay, there are other people who've shown other roles of other kinds of modifiers as well. Neurodevelopmental disorders. These are disorders that are, as written on the on the screen that you can read, right? Different kinds of autism spectrum disorders, movement disorders, learning disorders, communication disorders, intellectual disability overall of different sorts. And we know these kind of people exist, right? ADHD, etc., and people are born with these. These are called neurodevelopmental disorders because there were some defects that occurred that allowed the not normal differentiation of the neurons or oligodendrocytes, etc., to occur over time. Here is a summary of these. This is just shown as to what effects are due to what kind of cells, which you can see there are some of neuroepithelial cells, radial glial cells, etc., etc. Kabuki syndrome, number one here, is a syndrome caused by KMT2D, MLL2. This is a gene I study a lot in my lab. KMT2D is a histone H3K4 monomethyl transferase. What does H3K4 monomethylation does? Enhancers, thank you, sets up enhancers in your bodies, right? And it is absolutely crucial for gene transcription of large number of genes. That is where KMT2D mutations are found. Beautiful. Kabuki syndromes, people have problem in, this is one of the neurodevelopmental disorders. They have problems in, expressing themselves, speaking, etc., etc. Okay, read about this if you want. And then we talked about autism spectrum disorders, Down syndrome. We'll talk about Fragile X in a second. And there are multiple different kinds. ICF syndrome is caused by DNMT3A mutations, right? These are monogenic diseases, syndromes, meaning one gene problem. So when I say Kabuki syndrome, there's only one problem, KMT2D. So there's only one histone methylation problem, histone methyltransferase problem. Okay, got it? And what do monogenic, what is the significance of when I say monogenic diseases? Meaning there's only one gene. Cancer is a multiple complex problem of many, many genes, right? Whereas monogenic disease is just one gene problem, typically. Okay, got it? So that clearly says that if this gene was a problem, this syndrome occurred, that means this gene must be important. And if that gene has epigenetic function, then I will characterize this disorder as an epigenetic disorder. That's what I'm doing to you right now. Okay, I'm telling you that epigenetics is critical for many, many neurodegenerative disorders by telling you that there are mutations that cause these diseases. Okay, people have studied these in large sense and have found, extensive epigenetic dysfunctions, etc., in these disorders. Okay, and that makes a lot of sense. As I said, why I taught you transcription and differentiation, and I'll end with differentiation in the next class, you'll see, is because that is fundamental to many, many disorders, many problems that in itself, including cancer. I keep on saying cancer, but obviously these other problems are as important, right? Cool? Got it? By the way, Lee-Fraumeni syndrome, which is one of the major syndromes that causes cancer in the germline setting, is mutations of P53 gene, right? Cool? P53 gene is a tumor suppressor. Lee-Fraumeni people who are born with Lee-Fraumeni syndrome gets many different types of cancer as they go along. It's a transcription factor, right? We know it changes epigenome, changes many things, including epigenome, right? Again, things like that. So I'm just trying to sort of put perspective in your head as to think about how to think about these problems. ATRX syndrome is caused by ATRX mutation. What is? A chromatin remodeling machinery, as I showed you yesterday. Okay, multiple of these are just simply caused by epigenetic genes. Prader-Willi syndrome, okay, is a portion of the gene where a portion of the chromosome is, I think, doubled, and there is epigenetic function. We'll talk about Rett syndrome in a second, which is caused by MECP2 mutation, MEPCP2, that I told you has importance role in. Fragile X syndrome we'll talk about, and then so similarly there's more and others. I think Wolff-Hirschman syndrome is also an epigenetic gene. Okay, there's a gene called HDEC8 that I study, which is not written here, Carnelia-Delange syndrome, that is just by one gene, HDEC8, or cohesin mutations. Cohesin mutations, right? We studied cohesin yesterday. Yeah, cohesin causes 3D chromatin disorganization. Those patients also have mutations in this. Simple monogenic diseases, one gene problem, epigenetic genes. There you'll find a lot of such examples. And again, that makes sense because epigenetic makes the differentiation, right? Is the driver for differentiation, not the otherwise, right? Cool. So a number of these other things like epilepsy, autism, etc., people are studying the rules of epigenome and showing epigenetic differences. When you have to show causativity, what do you need? You need an engineered model, a genetically engineered model. People have made mutations in SMC, 3 and shown Carnelia-Delange syndrome like syndromes, Carnelia-Delange syndrome like phenotypes occur in mice when you make mutation in that gene. That's an evidence that that gene was important, along with the fact that patients who harbor this disease actually have mutations in that particular gene, right? That's how we demonstrate that an X gene is important for that function. Fundamental of science, right? Doing any experimental biology. Is that cool? Things like schizophrenia, things like Parkinson's, things like Alzheimer's, which are now neurodegenerative disorders. I won't talk about it, but there's a lot more literature coming it now, are not necessarily one gene problem. Okay, they are problems that we don't understand enough about. Okay, like Huntington is a Huntington problem, but these other neurodegenerative disorders where we see destruction of, like right, schizophrenia. There's a lot of in the different. They do not. That has been the puzzle. And if that changes, script, because clearly the gene is, but the same way. That is doing something else. Okay. That are in different neurodevelopmental disorders that I just listed, and on the right side are all epigenetic regulators that are mutated. So I was just not making it up for you, right? You can just go look it up and you will see that many of these genes are actually caused by epigenetic mutations. That's why the field of neuroepigenetics is very, very, very important for us. Okay, cool. So we are talking about learning, memory, which are our daily functions, right? We're talking about neurodevelopmental disorders where the embryos are born, we are born as individuals with those problems. Very different kind of phenomena. Then I also talked about neurodegenerative disorders that happen very late in life, beyond 80 years or 90 years as we live longer. That's a different kind of problem. So I'm just trying to set you up to think, don't get confused, because you'll get confused, and right? My habit is to mix up things, which is a problem sometimes. So learning is a different problem, memory is a different problem, has nothing to do with neurodevelopmental disorders, has nothing to do with neurodegenerative disorders. These are very different things in the neuronal world. Okay, all right? But the fact that you see these problems occur in mutations occur in epigenetic genes, the fact that you see epigenetic reprogramming occur in diseases tell you that there are underlying changes that might be important. And I use the word might be because you still need to demonstrate that those are critical. Okay, that's where again